Hello all of you. Today I would like to discuss about protein sequencing, which can be simply defined as the determination of primary structure of proteins. Since the functional structure of protein generally depends on the primary structure of protein, so it is very important to know the amino acid sequences in the primary structure of proteins. And the method through which we determine the amino acid sequence in the primary structure of protein is known as protein sequencing. It is a method to understand the structure and function of protein in living organisms. The primary structure comprises the identification of constituent amino acids with regard to their quality, quantity, and sequence in protein structure. So the first use of protein sequencing is to find the structure and function of protein. And the second main use of protein sequencing is to find the evolutionary history of a particular protein. This generally helps in understanding the changes over time in protein, shape, function, and its composition. So now I would like to discuss about different stages for protein sequencing. So there are three stages for protein sequencing. First is determination of amino acid composition in a particular protein. Second is degradation of proteins and polypeptide into its smaller fragments. And third is determination of amino acid sequence. So the second and third stages are interlinked with each other. So now I would like to uh, talk about all these three stages for protein sequencing one by one. So let's first discuss about the first stage of for protein sequencing, that is determination of amino acid composition in a particular protein. So in this stage, the protein or polypeptide is completely hydrolyzed to liberate the amino acids, which are later on quantitatively estimated. The protein hydrolysis may be carried out by acid or alkali treatment or by enzyme hydrolysis. Treatment of enzyme, however, results in smaller peptides rather than separate amino acids. But one enzyme, that is pronase, which is a mixture of non-specific proteolytic enzymes that causes complete hydrolysis of protein into its amino acids. So in this stage, whole proteins or polypeptides are used to hydrolyzed in presence of acids, alkali, which leads to its complete degradation into separate amino acids. Now these separated amino acids are generally identified by chromatographic technique, that is ion exchange chromatography, and by this way sequence or composition of amino acid in a particular protein can be determined. So this is the first stage of for protein sequencing. The second stage is the degradation of protein into its smaller fragments. So protein is a large molecule which is sometimes composed of individual polypeptide, or sometimes it is composed of more than two polypeptide. So separation of polypeptides of a particular protein is essential for its degradation, right? So liberation of polypeptides generally done by the treatment with urea and guanidinium hydrochloride, which disrupts the non-covalent bonds, that is hydrogen bond, hydrophobic interaction, etc., and dissociation into polypeptide units. For cleaving disulfide bond, performic acids are generally necessary. So this is all about the first step in the degradation of proteins into its smaller fragment, that is liberation of polypeptides by the use of urea or guanidinium hydrochloride, or and by the use of performic acid. So after that, when polypeptides have been separated, now we have to identify the number of separated polypeptides, right? So now we have to identify the number of separated polypeptide, and the number of polypeptide chains can be identified by treating the protein with densile chloride. It specifically binds with the N-terminal amino acids to form densyl polypeptides, which on hydrolysis yield N-terminal densyl amino acids. So the number of densyl amino acids produced is equal to the number of polypeptide chains in a protein. So since here two N-densyl amino acids have been formed, that's why the total number of polypeptides here are polypeptides in a protein are 2, that is polypeptide 1 and polypeptide 2. So the next step in this direction is the breakdown of polypeptides into fragments. Here polypeptides are degraded to small peptides by enzymatic or chemical methods. So in enzymatic cleavage, polyproteolytic enzymes such as trypsin, chymotrypsin, pepsin and elastase exhibit specificity in cleaving the peptide bonds. Among these enzymes, trypsin is most commonly used. It hydrolyzes the peptide bonds containing lysine or arginine on the carbonyl side of peptide linkage. In chemical cleavage, cyanogen bromide is commonly used to split polypeptide into its smaller fragments. Cyanogen bromide specifically splits peptide bond at the carbonyl side of which is contributed by the amino acid methionine. Now the third stage is the determination of amino acid sequence in a particular in all these short peptides or all these polypeptide fragments, like, and this stage and this process are generally governed by two methods. First is Sanger's method and second is Edmond degradation method. So I will explain all these I would like to talk about these two methods of protein sequencing one by one. So let's come first on the Sanger's method of the determination of amino acid sequence in a particular peptide or polypeptide subunit. So let's first talk about the Frederick Sanger. And Frederick Sanger was a British scientist who got Nobel Prize twice for chemistry. First Nobel Prize he got for his discovery of the structure of insulin molecule in the year of 1958, and second Nobel Prize he got in the year of 1980 for his collaborative work on base sequences in nucleic acids. So by this you can understand about his immense contribution in the field of chemistry and molecular biology. So in protein sequencing, Sanger's used one compound that is known as 1-fluoro-2,4-dinitrobenzene, and in short we can say it as FDNB, right? Here FDNB specifically binds with N-terminal amino acid to form dinitrophenyl derivative of peptide. This on hydrolysis yield dinitrophenyl amino acid, right? And rest of the this on hydrolysis yields dinitrophenyl amino acids, which is generally present in the N-terminal of amino acid, and side by side it also produces free amino acid from rest of the peptides, right? And this DNP amino acid can be identified by chromatographic technique. So you will have to repeat the whole process for identifying the next amino acid in peptide. So the main drawback of these processes are you will have to repeat the process again and again to know the complete sequence of a particular peptide or polypeptide. and efficiency of this process is quite low. That is, peptides with only 60 amino acid residues can be sequenced properly by this method. So these are the two drawbacks, and these two drawbacks are generally mitigated by the discovery of or the invention of other method of protein sequencing, that is Edmonds degradation method. So Edmonds degradation method of protein sequencing was developed by Fur Edmonds, and it is a method of sequencing amino acids in a peptide. So here phenyl isothiocyanate is the Edmonds reagents, and it reacts with phenyl isothiocyanate generally reacts with N-terminal amino acids of a peptide to form phenyl thiocarbamyl derivative, and on treatment with mild acid this phenyl thiocarbamyl splits into shortened peptide and and phenyl hydantoin amino acids, right? And these amino acids were further identified by chromatography technique. So in this way, in a single reaction, in Edmonds degradation method of protein sequencing, we can determine the whole amino acid sequence of a peptide or a polypeptide, and efficiency of this method for protein sequencing or amino acid sequencing in a particular polypeptide is also greater than in comparison to the Sanger's method of protein sequencing. So this is all about the different method of the protein sequencing, that is Sanger's method of protein sequencing and Edmonds degradation method of protein sequencing. So now I would like to discuss about the denaturation of amino acid. The phenomenon of disorganization of native protein structure is known as denaturation. And here denaturation results in the loss of secondary, tertiary and quaternary structure of protein. This involves a change in physical, chemical and biological properties of protein molecule. So there are two terms. First is denaturation, second is degradation. So in degradation, the hydrolysis of peptide bonds generally takes place, but in denaturation, the hydrolysis of non-covalent bond and disulfide bonds generally takes place. Like here, disulfide bond is also a covalent bond, and non-covalent bonds are, or you can say it as weak bonds, are hydrogen bond hydrophobic interaction, van der Waals force are the weak bonds. So the agents which are used for the denaturation of a particular proteins are physical agents and chemical agents. So heat, violent shaking, X-rays, UV radiations are the physical agents, and acid, alkalis, organic solvents, salts of heavy metals, urea and salicylates are the chemical agents. So now I would like to talk about the characteristics of denaturation. So in denaturation, the native helical structure of protein is lost, and the primary structure of a protein with peptide linkage remain intact. That is, peptide bonds are not hydrolyzed. So in which process peptide bonds are generally hydrolyzed, and that process is degradation of protein. In this process, the protein loses its biological activity. Other important characteristics of denaturation of protein is that denaturation is associated with increase in ionizable and sulfide group of protein. This is due to loss of hydrogen and disulfide bonds. Denatured proteins is more easily digested. This is due to the increased exposure of peptide bonds to enzymes. Cooking causes protein denaturation, and therefore cooked food is more easily digested. Another important point or characteristics of denaturation is usually irreversible. For instance, omelette can be prepared from an egg, but the reversal is not possible. But there is one exception of this statement, that is, careful denaturation is sometimes reversible, known as renaturation, and hemoglobin undergoes denaturation in the presence of salicylate, but by removal of salicylate, hemoglobin is renatured. And the last characteristic of denaturation is denatured protein can be crystallized, cannot be crystallized. So what is the significance of crystallization of protein? Actually, crystallization of protein is generally used for deriving the structure or identifying the structure of the native protein, that is functional protein. But since in denaturation of the protein we generally disturb the native structure of protein, that's why denatured protein cannot be crystallized. So this is all about the chapter protein, and if you feel any problem in understanding anything regarding this chapter, you can call me and you can message me. So thanks to all of you, and bye bye.